Thank you everyone for being here, and welcome to the ALS Center Symposium for 2026. I'm Dr. Sean Smith, if I haven't met you before. Just to start, a huge thank you for the whole team that put this together. It requires a lot of hours, and just to highlight a couple of people, Kelly McCoy-Gross, and Erica Jacquez, and Kate Moran for their dedication and making sure that this gets up and going each year. And let's start out with reviewing the agenda that you all have. So we'll do our welcoming, and I'll do some opening remarks, and then Dr. Tim Miller will be doing the keynote address. And then we'll have several presentations with the lab and clinical perspectives. And one by Dr. Ravi Chopra, Can We Catch ALS Before It Starts? And then Paige Kirshner, PhD, on TDP-43. And then Dr. Dylan Galloway with blocking small MRAs to protect motor neurons. And then Ashwara Nambiar, PhD candidate, we'll be talking about protein boosting ASOs as a therapeutic strategy for TBK1 ALS. And then Monique Costello, our social worker, will be talking about adapting to life with ALS. Then we'll have a break so everyone can get a little bit of a rest and refreshments. And then we'll come back, and Amber Malcolm, co-director of the ALS Clinic, will do a patient and caregiver perspectives panel. And then Dr. Bobbicelli will be doing the audience Q&A session, and followed by the recognition of research participants and closing remarks. And then we'll have our lunch reception. So for Q&A, for in-person questions, there's index cards provided in the folder. So please write any questions on those, and the staff will collect them during the breaks. It would help if you were able to pass them down to the end of the aisle, and then we can pick them up. For Zoom attendees, you're in listen-only mode, and you can type questions at any time via the Q&A feature, and we'll answer the questions during the Q&A portion of the agenda. And the red arrow sign is showing where the Q&A is on Zoom. And questions are only visible to staff and will not read your name out loud when answering. A couple of housekeeping items. Parking, pick up a garage voucher at the registration desk. Restrooms are located on the first floor, are regular bathrooms. Accessible and assisted bathrooms are on the second floor. There is a lift if anyone needs one. If you have any questions, please ask one of us and we'll help you. This event is being recorded and photographed. We plan to publicly use, if we publicly use any images of you, we'll contact you for your consent before doing so. And please sign up for our newsletter. The sign-up sheet is at the registration table, or you can go to our website. And then please do follow us on social media, Twitter or X account, and we also have a Facebook. And I just wanted to highlight in this spring-summer edition of our newsletter, there's a great article on Laura Rocker's charitable foundation and partnering with ALS research and all the contributions and fundraising that goes to support the research at the Miller Lab. So some opening remarks, a short presentation. It sounds much more in-depth than what I'm going to cover, but transforming ALS care, at-risk presymptomatic programs to diagnosis and recovery, and just kind of highlight the research that we've been doing. And here at the WashU ALS Center, we're building a future for the next generation of multidisciplinary ALS care. And that's through research with advanced diagnostics, research on disease-stabilizing treatments, integrating neuromuscular rehab, excellence in ALS clinical care, and we are an internationally recognized ALS research program. Very hard to read, so I don't want you to do that. I just wanted to highlight we initiated an ALS at-risk program for individuals with genetic forms or genetic variants of ALS who have not had symptoms yet. And so part of that is doing the genetic testing. We do presymptomatic biological disease monitoring. You'll hear about a protein called neurofilament light chain. And then people have the opportunity here to participate in ALS, Prevent ALS clinical trial, which monitors patients over long periods of time, participants over long periods of time, to see if we can learn about when ALS develops, how it develops, can we catch it earlier, and then can we intervene before someone has symptoms. And Dr. Bob Buccelli is the site investigator for the ATLAS trial, which is exactly doing that in SOD1 ALS in the use of tofersen. And so for advanced diagnostics, a study that I took over from Dr. Buccelli here, led by colleagues in Chicago, is looking at using a type of neurodiagnostics called intermuscular coherence to diagnose ALS earlier. And so there was a publication recently on primary lateral sclerosis. And earlier last year, a publication on using that diagnosis with ALS. And then more recently, our team published a paper on integrating the use of this year of neurofilament light chain to improve the diagnostic certainty around ALS. And so, as you can see, we are advancing that field. And then I also wanted to highlight who's not here today, Dr. Cindy Lee, who runs the postmortem tissue research here. And she also is doing multiple studies into looking at other biomarkers to help diagnose ALS sooner, and then also in the treatment response. Dr. Josh Newman wrote a comprehensive review on SOD1 ALS and targeted therapies. You'll hear a lot about tofersen today. And then Dr. Miller, who led the studies on tofersen, the recent long-term follow-up that was published earlier this year. And then we have our tofersen clinic. So it's precision medicine using tofersen, which targets SOD1 protein. It's a comprehensive, integrated, multidisciplinary care clinic. Some of our participants do the neuromuscular rehab. And so it's been, we're very grateful for our participants because they not only come and get treated with tofersen, but they participate in about 5 or 6 other studies. This is a new era in ALS care and treatment in that we previously never had people who had their disease stabilized. And so just the time, commitment, and the dedications that we can learn from you, is really touching. And it's the only way we can learn. And so we are helping to advance that field. And our study, Rehabbing SOD1 ALS, is providing fundamental knowledge on ALS recovery, what we have to do, what it looks like, how we can maintain function, and how we can improve function for individuals. And I wanted to highlight my colleague, Dr. Beth Hughes, who's a physical therapist at WashU PT. She's in the ALS clinic with us on Tuesdays, and she sees our patients on tofersen, and she has really instituted neuromuscular rehabilitation program. And we are the first to publish on our program, and look forward to continuing this work with Beth. And that we did publish a paper recently on our experience, and so we'll see, we're instituting what we're calling a new model of care for ALS. And it's based on the rehabilitation that's done in spinal cord rehab and traumatic brain injury, where there's multiple sites that do research It's kind of hard if you don't have a large population of individuals, but to gather that data together and hopefully find best practices to be able to guide in this early stage of clinical stabilization in ALS to guide people in their rehab. And this is a diagram from that paper, and what it shows is, at least for our individuals with SFD1 tofersen, is that there's ALS progression before diagnosis, right? Including diagnosis, and then when patients start treatment with tofersen, it takes a couple of months for tofersen to kick in, and so we do continue to see some progression. Over a period of months, probably around 6 months to 9 months, we start to see some disease stabilization in patients. And at this point, we've started instituting the neuromuscular rehab early at the time when individuals start tofersen. And what we see is that there's a possibility that it helps not only stabilize disease, but improve function during that time. And then long-term, continued neuromuscular rehabilitation, thanks to Beth's work. We've been able to identify different phases to progress individuals through who are doing the neuromuscular rehab. And what we see is that although patients don't have a full recovery, there is improvement in function and functional independence. And this is unheard of 5 years ago in ALS. And a big thank you for our excellence in ALS clinical care to our clinic team on Tuesday. This is our 2-year picture. We're at 3+ years. We haven't taken our recent picture yet. So, and these are our friends that come and visit us to help lift up everyone's spirits. And then this is part of the research team and part of the clinical team at the ALS Walk this year. This is, I'm holding George, and then there's Duke. They like supporting the ALS Walk. And like many of you who did the walk, I have a family member that has ALS, and his name is PJ, and he was diagnosed when he was 28 years old, and today is his 30th birthday. So happy birthday, PJ. So what you hear today is what makes us an internationally recognized ALS research program. And everything is guided by our mission, which is to deliver high quality, compassionate, and comprehensive ALS clinical care, and to pursue cutting edge research focused on finding new therapies for ALS. And so I won't go into that any further, but you'll get to hear some great talks today. So bring up Dr. Tim Miller, co-director of the ALS Center, and he'll get us started. Thanks.
Okay, thank you, Sean. That was a fantastic start. Can everybody hear me okay? Yeah, all right, very good. I also want to welcome everyone and thank you for being here. Please do fill out these cards with questions along the way, or if you're on Zoom, fill out some of the questions to go. We're going to have a time period for questions. I do always start with my industry collaborations, in particular Ionis Pharmaceuticals, with whom I've had the pleasure of developing antisense oligos, and I'm going to talk about some of those today, and Biogen, with whom I've had the pleasure of developing clinical trials, running clinical trials, and I'll talk about one of those today. And I realized this morning that I should have updated this BIO, which is no longer on there, but Arbor Bio, just to be 100% current. I also want to give a thank you to those that really helped to put together the symposium. It was our whole group. We meet weekly, and this was a discussion topic weekly for months on end in terms of how to deliver the symposium to you. Jesse and Kelly really leading the charge, but huge input from Erica and Kate, Amber and Sean, and again, really the entire group. I do want to introduce the concept of the ALS Center. Bob Uccelli, who's sitting up front, is co-director of the center. And we think about ALS clinical care, clinical trials, and then basic research. And we try to bring all of those together to find new therapies for ALS and also to deliver outstanding care. The next couple of slides are going to go by quickly with some of the names and pictures of the people that do the work in this center, but I do want to highlight and recognize them. many of you are taken care of by many different providers at WashU. So it's not just a few of us taking care of ALS. Our team, the whole team takes care of ALS, and you can see all the different people that are listed there. And then we have the motor neuron collaborators not taking care of ALS, but focus on spinal muscular atrophy, so an important collaborative group in pediatrics. And of course, clinical care for ALS is not just the providers, but also the rest of the team, and is shown here. And I won't go through, again, each of the people here, but nurse, dietitian, and physical therapists, occupational therapists, and others that are focused on delivering really top, high-quality care. Now, those are the people that you will have seen. You may not have seen some of these people. They're in the back, basically the back offices, doing the work to get the things moving forward, authorizations for equipment that you may need, or medications, and that's this group here, who also are really fantastic. You may have interacted with some of these people on my chart or by email. Just some of the stats for our ALS clinical care: more than 150 new patients seen per year. That's more than 350 return visits, so a lot of people being seen here, and it's a full, comprehensive clinical care. And we serve 300 miles around St. Louis, and even further than that, but people really do travel to come to St. Louis for the clinical care. And we were one of the first groups to launch a newly approved genetic-focused therapy, tofersen, which I'll talk about. We've been able to launch the new thing actually multiple times for new ALS. This is the group doing clinical trials. Of course, it overlaps with the group doing clinical care, especially on the top row. There are many of us involved in the clinical trials. And then you can see down below, led by Jesse Markway, the clinical research group. And if you're in a clinical research study here, some of these names may be very familiar to you, people running these clinical trials. Some of the stats in our clinical trials: a top enrolling center for clinical trials. We only take on the trials that we think are important and that we think will be interesting for the community here. And by doing so, we're typically a top enroller in those trials. People want to sign up. We've done, and I should have checked with Dr. Bocelli about the numbers here, but more than 900, maybe even more than that now, LPs completed here for the antisense oligo or ASO trials alone. We typically are doing more than 10 studies, and I'll show you some of those studies, some of them active, some of them no longer enrolling, some observational, etc. We launched one of the largest ever natural history and biomarkers study, all ALS. And this group here is guiding the national conversation in ALS research. As part of the executive committee for the Healy platform trial, as the co-chair and a member of the scientific advisory board for NEALS, or the Network of Excellence for ALS. And I'm a former co-chair of NEALS and remain on that leadership group, so very involved in those clinical trials. Highlighting the clinical trials now at WashU, there are 3 trials open and enrolling. COYA, which is a phase 2 trial, PREVAILS, a phase 3 on pridopidine, and tofersen in non-SOD1 ALS, and I'll come back to tofersen in just a bit. But there are also observational studies. Nearly everyone in this room could participate in an observational study, because we have opportunities for people with ALS or healthy controls, gene carriers, etc. And we are very active in these studies. And I've listed as well those that were active but not currently enrolling. So these are studies that still ongoing, very active in terms of people coming in and being seen as part of that study, but we're not enrolling new people. I do want to take a second here to remind you that we have posters today describing some of these clinical trials and describing some of our work. So during lunch or during the breaks, please feel free to stop by those posters. We'll have the people there hopefully be able to explain that to you. Okay. So we also do basic and translational research. And I've listed here my group, Cindy Lee's group, and Kathleen Schuck, who's really integral to my group. It's really the founding members of the ALS clinical basic and translational research at the ALS Center. But as I have integrated, more and more over the last decade into WashU, I've recognized that there are many, many other groups doing ALS research, and I've listed them here. Some of them are here today. I think I saw Eric Arnold, Laura Capizzi, and others that are here. There's a bunch of groups focused on ALS. Many of these that we are in one way or another collaborating with. Many of these people using the resources from the ALS Center, for example, biofluids, postmortem tissue, et cetera, that are shared with the world, but also shared here as well. These, I said updated soon. Actually, some of these are already updated on the ALS website. So look for, there'll be a tab that says basically partners and labs, where we will link out to and list each of the people at WashU doing some sort of ALS research and link to their work as well. Highlighted in the Miller-Lee lab, some of the people that are doing the work. You're going to hear from some of those today. That's new this year, asking some of the graduate students and postdocs in the labs to present some of their work to you. This is actually the hardest talk that I've asked any of these people to give this whole year. You say, well, why? It's only 5 minutes. It's to a very friendly lay audience, but it's difficult. So it's difficult to communicate the things that we do to the lay audience. I know that they've given it a lot of thought, but if you have questions, fill them in as we go. All right. Some of the things that we're doing, ways to turn off harmful genes, SOD1, I'm going to describe that a bit more. We're developing a new method to increase a gene product, and Aishwarya is going to talk to you about that. Understanding why some motor neurons are protected, not others. I don't think we have a talk on that today, but a very active research program led by Dylan Galloway. As many of you know, the eyeballs continue to move just fine in ALS. Eye movements are not affected, but those are motor neurons, the same cell type that controls movement of, for example, your arms and legs, which are usually affected in ALS. Why? We still don't know, but there are new methods and tools that have come through to help us understand that. We're looking at large data sets. I don't really do data science, but recognize the need to do data science, and we've got some great projects going there. We're also developing ways to degrade harmful proteins. You'll hear a little bit about that today, and developing new biomarkers. And I want to recognize, as Sean Smith already did, that our new ideas in the lab are often supported by philanthropy. Like, how do we get started for for doing something before we can write that very large public NIH grant? It's with the new things are funded by philanthropy, and I'll give a special shout out to the Lyle Rockers Foundation. I see Lyle up there. Okay, I'm going to talk to you about tofersen, and to understand this drug and the way it works, you need to understand a little bit the genetic landscape of ALS, what we call 90% singleton or 10% familial. Singleton is a fancier term for sporadic. Most people say sporadic, and I probably will say sporadic throughout the rest of this talk. Singleton really means that there's one person in the family who's got ALS, but not anybody else known to have ALS. But yet they still may have a genetic cause, and that's what we found. So we would call that genetic ALS, not sporadic. Okay, in that 10% that's familial, there's a gene called SOD1, or superoxide dismutase 1. This was the first gene that was linked to familial ALS. And it's 10 to 20% of familial. If you do the math, it's 1 to 2% of all ALS. Actually, the numbers are probably closer to 2% of all ALS. It's about 350 people in the United States living with this disease. Superoxide dismutase 1 causes about 2% of ALS. It's a genetic change, one letter that's changed in the DNA. It's typically not associated with dementia, and it causes toxicity to neurons from this abnormal protein that builds up. And I'll talk about that a little bit. Tofersen is designed to lower the SOD1 protein, designed to treat ALS for those with SOD1 mutations. Okay, so I'm going to take you back to high school biology. Some of you have seen this before. We're going to talk about DNA, RNA, and protein. All right. And so we start out with DNA. DNA is like a set of instructions, a blueprint for how to make a human body, how to maintain a human body. But it's complicated, and it gets rewritten into basically another set of instructions. If you're thinking about building a house, maybe this would be a set of instructions for just a living room, So that would be your RNA, a rewritten pamphlet, different set of instructions. And you think of the proteins, that might be the stuff in the dining room or living room, so the rug, the chair, things like that. Or perhaps, I'm showing you here, a brick, and we'll come back to the brick. Okay, so you got DNA, RNA, protein, and you have the, shown here, you have a change in the genetic code, which leads to a change in the RNA. If you rewrite that set of instructions, still have the same mistake there. And then that leads to a buildup of the toxic protein. Okay, so you have this set of instructions, which is DNA, and it gets rewritten into another pamphlet. Both of them have a mistake in it. And that mistake gets followed through to this brick that looks okay for now, but actually, as it turns out, it's got a mistake. And so it's not a regular sort of brick, it's a misshapen sort of brick, which does okay for a while. As you might build a house, it looks okay at first. People with these genetic mutations, ALS, they're fine. But over time, there's wear and tear, things begin to break down, and eventually the whole thing falls apart. And that's the way that I at least think of the genetic changes associated with ALS. Antisense drugs or antisense oligonucleotides, one of them called tofersen, block these disease-causing RNA. You can think of it as an eraser that blocks the RNA, takes out that specific set of instructions, so it's no longer there. And if you block that RNA, then you block the protein. And I'm going to show you some of these results from VALOR, which is a clinical trial, a phase 3 clinical trial, 72 people on the drug, 36 people on placebo, 3 doses of the drug given in the first month, and then 1 dose of drug about once a month after that. These fancy RNA blocking, RNA erasing drugs do not cross the blood-brain barrier. You can't give them in the blood. Well, you can give them in the blood. They just won't get into the brain, won't get into the spinal cord. We know that from years of studies. So this is delivered intrathecally. So your spinal cord ends, I'm going to try the laser pointer. Your spinal cord ends about 2/3 of the way down the back, and then there's this fluid-filled space where there's a bony column but no spinal cord, and you can put a needle in there and withdraw fluid. Many of you will be familiar with a lumbar puncture. Or you can actually withdraw a little bit of fluid and then push fluid back in. And then as shown with these blue sort of dots, this antisense oligonucleotide then distributes throughout the brain and spinal cord to deliver this drug to all the places that it needs to go. And I'm just going to show you a few of these results. This is the strength testing from this trial as reported out at 3 years. And importantly for this study, at 28 weeks, everybody goes on the drug, called the open label extension. People that get the drug early here, people that get placebo in both groups, about the same at 28 weeks. And this is what you'd expect to see in terms of the projected course of ALS. And instead, this is what we saw. A flattening, a bending, a change in the curve where instead of relentlessly, progressively downhill, stabilization. To get this sort of stabilization, especially out of 3 years in an ALS clinical trial, some people ought to stabilize completely. And we had heard rumors of people actually improving in terms of their strength. So we looked at this at 148 weeks, nearly 3 years after the beginning of this trial. And we looked at 3 things: ALS functional rating scale, breathing, or slow vital capacity, breathing, handheld dynamometry, strength. And then we looked at it in terms of fast and slow progressors, and this really has to do with the biomarker neurofilament. I won't go into many details there, but we had a fast progressing group and a slow progressing group. And I blanked it out, usually because in many audiences, maybe I don't need to tell this audience, what we expect to see at 3 years is that nobody would have stabilized or improved, unfortunately. We just wouldn't expect to see that, not in a disease like ALS. And instead, in this study, what we see, about 20 to 25% of people overall have stabilized completely or improved out at 3 years. And in the slower progressing group, really impressive numbers in terms of improvement in strength when we look out at 3 years. And we've heard many, many anecdotes, perhaps some from people in this room and their families, but really from around the world, about people who have stabilized or improved on tofersen. It's not without side effects. I always present this slide: myelitis, radiculitis, papilledema, some of these medical terms, perhaps not important for me to go through, other than to say that 8% of people on the drug do get some serious side effects. We have to take that into account. Okay, some conclusions. Tofersen lowers the levels of the biomarker and substantial slowing of neurodegeneration. It's a 3.4-year increase in survival. I didn't present those data today, but that's nearly a doubling of the expected survival in this patient group. And then I already mentioned the stabilization and improvement. The drug was approved by the FDA here. So the first approval was by FDA in 2023, then in Europe, and now, as of February 2026, when I last checked in on this, in 44 markets around the world. I want to be clear to thank the many, many groups involved in this. I don't have enough time in this talk to really thank the people who have contributed to this over the 20-year journey of developing this drug. But I do want to in particular highlight the participants and their caregivers and families. People ask, was this difficult to get the LP? And perhaps some of you can comment on that in this room better than I. But in general, that was not the hard part about being in this study. The hard part about being in this study for many people coming hours away to get to St. Louis for this drug was the travel here. Many people are aware that when you have the disabilities associated with ALS, it's hard to get here. And they needed to get here about once a month. There were many people in the steering committee involved, the principal investigators and colleagues at Biogen that I also want to recognize. And then, how does tofersen relate to sporadic ALS? Because many of you will think that's a great story, but how does this relate to the thing that I have, which they told me I don't have genetic ALS? So how do we link that in? And before I start, I also want to recognize that I've been super excited about 20 to 25% of people completely stabilizing and you're getting better. And part of our work here, part of our thinking here is that that's not good enough. It's fantastic. It's actually a miracle that we're able to have that kind of effect on ALS at all, but it's not good enough. We want to see everyone treated with tofersen who walks in the door to be completely stabilized or in fact improved. And so that's where we are headed. And then pivoting to sporadic ALS. What about sporadic ALS? I think this teaches us that ALS is a treatable disorder, which is a big deal in the ALS world. A big deal for the academic community. We've seen so many trials fail. Are we ever going to get one that works? If we get the right drug, we can get one that works. It's a big deal for the pharmaceutical community, who had a little bit lost hope that they could ever find something. It might have been true that once ALS starts, there's no changing it, right? Yes, you can put out the match, but you've started the fire and it's already going and there's nothing you can do. And we know that that's not true now. We know that if we get the right drug, that we will be able to treat ALS and change the course of ALS. And of course, we're working on all sorts of ways to get the right drug to do that. The biomarker change. This is also a big deal in the ALS world. 60% lowering of this biomarker in blood teaches us how to do trials, teaches us what we can look for early on. 6 months, we didn't see much of a clinical change. We saw a massive change in that biomarker out at 6 months. And that ability changes the way that people are running clinical trials. All trials will include neurofilament. Some trials will look just at 6 months and get an early readout. Is this a good drug or not a good drug? And that just increases the throughput, the ability to take shots on goal, and changes the way we're doing clinical trials. What about genetic therapies for sporadic ALS? This is sort of an out there idea, that we developed a fancy new therapy for FUS, SOD1, CNR72, all the genetic changes associated with ALS. Maybe we could use that for sporadic ALS. What's that link? And I'll tell you that we've recently launched a clinical trial of tofersen, which is for SOD1 ALS, as I've described it, in non-SOD1 ALS. And if I had another 40 minutes with you, I'd be able to go through that in more detail, but I won't today, other than to say this is a concept that we are now embarking on, and I guess we'll see how that goes. And then the last thought is ASO delivery to the CSF can be effective. This was doubted for 20 years or so, that we could do this, that it would have an effect. We've now shown that it has an effect. And there are many follow-on studies using the exact same, I call it a blueprint, that we have put forward for how to treat ALS with these therapies delivered to the cerebrospinal fluid, this intrathecal delivery that delivers it to the brain spinal cord. Okay, I want to thank you for your time. I do have one more a brief 3-minute video. I'm pausing here. If you wanted to grab the ALS Center site, you could grab it here, or just alscenter.wustl.edu has a lot of information about our center, and we try to keep that updated, and we'll be adding more and more information about ALS at WashU. And I'm now going to share a brief video with you, produced by colleagues here at WashU, so the WashU Media Group, focused on tofersen. And then I'll be done handing it off to the next speaker, hopefully close to on time.
In the United States, there are about 30,000 people living with ALS.
You know, one of the first things that pops up, life expectancy is 3 to 5 years. You know, then that's in the back of your mind all the time.
The ride home, we were both just silent. We were almost grieving someone that's alive.
My boys were confused. My wife took it really hard. I took it hard.
By the time I was diagnosed with ALS, I was 23. You're kind of told you have 2 to 5 years to survive. Yeah, it's just shocking.
I was probably 2 or 3 when I asked the question, and I was told ALS is how we grow up in our family.
Finding out that I had it was almost relief, because it was like, okay, the monster has a name, I have a goal.
I didn't expect to be here at this point. I surpassed the expectations everybody had that I wouldn't survive, and to do it able-bodied is miraculous.
Trey's more active now than he's ever been. He can do everything on the farm now that he did 5 years ago. If you didn't know him, you wouldn't know he has ALS.
There is so much hope right now. Your life's not over. I'm living proof of the research. Like, I'm still here, I'm thriving, and I should be dead.
ALS has been a mystery for 150 plus years. Never before in the history of the treatment of ALS have patients improved over a period of years. It's a new era to ALS treatment.
She actually said, this is moving from trying to prolong your life to trying to save your life.
So many people just never thought this was possible. Single-handedly the coolest thing that I've seen and experienced in my career, and hands down the most rewarding thing.
People are stronger. Yes, we are reversing some parts of ALS. It goes against the natural history of the disease. It goes against everything we've come to understand about ALS.
I said, when I got this, that I was gonna fight for years, months, days, hours. Because I wanna be with my kids as long as possible. And thinking about the fact that I might get to see my daughter get married. That's everything to me.
They continue with this relentless commitment to moving things forward. How can you not be inspired by that?
Okay, thank you. A really moving set of stories. I want to recognize the WashU Media Group for putting that together, and of course all the people that involved in that video and overall the tofersen program. I'm done. A few questions, write them down, put them in the chat. I want to recognize and thank the Zoom audience today. Thank the media crew here for setting that up. And now welcome Ravi Chopra to kick off, I think, our lab and clinical perspectives. There he is. Thank you.